Good evening, everyone. Oh, wow, that is very, very loud. Thank you so much for coming. My name is Caleb Weddington. I am the president for the University of Shadysboro American Society for Biochemistry and Molecular Biology. We have a very special guest this evening, Miss Doctor, I should say, Lauren M. Oldfield. She received her bachelor's degree in microbiology at the University of Akron. She received her PhD at the University of Pittsburgh. She is currently a synthetic biologist, a microbiologist, and molecular biologist with the J. Craig Venter Institute. And for those of you unfamiliar with the J. Craig Venter Institute, they are the ones who brought the full human genome sequence to the world, which was a monumental and groundbreaking event. And I say that because a groundbreaking institution attracts groundbreaking individuals. We have one here with us tonight. She is an expert on viruses, and I am quite confident that she has quite a lot to say to us. So without further ado, please give a warm welcome to Dr. Lauren Oldfield.
Thank you. Thank you. I won't use the mic, so if anybody has problems hearing me, let me know, but I'm used to talking loud. So I'm going to tell you a little bit about how I became a scientist and the things that I studied and the pathway that I used to get here. But I'm going to kind of do this all in reverse order because I'm going to talk a little bit about the J. Craig Venter Institute and I'm going to give you guys some of the actual science that I've been doing. And then we can do more of sort of like an open discussion. Please feel free to ask me questions. And we'll talk more about like how you get a PhD and what that's like. But definitely feel free to stop me at any point if I've confused you or if you have questions. I like this to be more informal and it makes it easier for me too. So the J. Craig Venter Institute we've got two campuses, one in La Jolla, California, right outside of San Diego, and one here in Rockville. And it's literally right around the corner from here. So it's like a 5-minute drive. We're right by the hospital off of Shady Grove. And we're a not-for-profit research organization doing lots of work in genomics and microbiology, lots of different fields. We were founded originally in 1992 as the Institute for Genomic Research. And a bunch of smaller institutions, including Tiger, merged together in 2004 to 2006 and became the J. Craig Venter Institute, which I'll just refer to as JCVI throughout the talk. So that's what I'm talking about. And the overall goal of the Institute is to focus on science that improves human health and the health of the environment. So some of the work that we've done at JCVI, we published the first sequence, full-length sequence of a free-living organism, which is Haemophilus influenzae, which is a bacteria. And we went on to sequence lots of microbial genomes, including some of the things I'll talk about today. We're doing lots of viral sequencing now. JCVI with the team at Celera, which was a for-profit company that Craig Venter also was in charge of, sequenced the human genome. And they were actually in a race with the NIH and the Human Genome Project. So they were privately funded, and the NIH funded their own project to sequence the human genome. And the picture on the top over there is the Time magazine cover when they published the human genome. So the two sequences actually came out at the same time. We've also done lots of metagenomics, which is basically going out into the environment and collecting samples and sequencing whatever is there without actually isolating any organisms. And so one of those was the Global Ocean Sampling Expedition, and you can see the boat that they used. And they've literally traveled the world in this boat now. And they have special scientific equipment on board, and they collect water samples throughout the oceans and sequence all of the organisms that are present, including things you wouldn't be able to see otherwise because they're probably not culturable, or at least we don't know how to culture them in the lab yet. And the synthetic biology group at JCVI, this is sort of the most recent thing, came out with the synthetic genome of an organism, which is a mycoplasma, which is a very small bacterial genome. So that's the SynBio department that I'm a part of. So JCVI is ranked in the top 1% of research institutions, and this is basically based on citations of papers. So it's kind of an antiquated measure. I don't know that it really shows the full range of science that a particular institution does, but a lot of this is based on a few of these major like landmark scientific papers, including the microbial genome that I talked to you guys about, the human genome, some of these environmental genomics projects like the ocean sampling, and we're also in one of the institutes that was involved in the human microbiome project, which has to do with sequencing all the bacteria that live in and on you as a person. And so there are 5 main groups at JCVI. I am in the synthetic biology and bioenergy department, although I don't have any experience in bioenergy, but there are other professors in my department that work on that. And a lot of the stuff I'm doing now actually sort of bleeds over into the infectious disease group. But we also have genomic medicine, which is based more on human genetics and looking for human disease-causing alleles. This microbial and environmental genomics, and then there's a group that also does plant genomics. So we're based, you can see a lot around sequencing and the synthetic biology aspect of then like rebuilding those sequences. So then I'm going to move a little bit into the science that I've actually been working on. So all of this happened before I was a member of JCVI. I've been there for about 3 years now, 2.5 years now. And so my project isn't any of those things that I was telling you about, but I've been working on viruses. So the most basic definition of a virus is that it's nucleic acid in a protein coat. So there's some sort of genome, either RNA or DNA, and it can be either single-stranded or double-stranded. And that's encapsulated in some sort of protein that surrounds it. Viruses don't encode ribosomes. At this point, that is true. We haven't found a virus that encodes a ribosome yet, but you never know what we might find eventually. And we think a lot about viruses that infect us and cause disease. But there's lots of viruses out there that aren't infecting us. So genomes range from being very small, so 1,700 base pairs, to being over 2 million base pairs, which is larger, and they're physically larger than some bacteria, these largest of the viruses that we've seen. They can have anywhere from like 7, these small viruses have like 7 genes, to over 2,000 genes, and their physical size of the particle can be very different too. So this gives you an idea of, this graphic over on this side shows you the flu virus, which is a pretty standard size virus. And then some of these larger viruses that infect, mostly these super large viruses that we've found have been, they infect amoeba. For some reason, all of these very large viruses that we know of so far are amoeba viruses, and the top picture is an actual electron micrograph of the virus, and they've colored it, so it's falsely colored, but that shows you what the virus really looks like. And then there's these interesting satellite viruses, just to give you an idea of sort of what's going on. They can be even smaller. And what do viruses infect? They infect everything. Most of my PhD work was spent studying a virus that infects bacteria, so I'll tell you guys some about that. We call them bacteriophage. And so this first picture is a picture of bacteriophage mu. And you can see they're really cool. I'll show you some pictures of like some artistic renderings of what they would look like. But they basically look like little alien landing pods with tails, and they have tail fibers that go down and touch the bacterial surface. There's plant viruses, animal viruses, including viruses that infect us, obviously, like the flu virus. And I like this picture of tobacco mosaic virus. It's really cool because it has this really interesting long rod shape that I don't know that very many viruses have. So like I said, I worked with bacteriophage in my PhD in the lab of Graham Hatfull at the University of Pittsburgh. So this is the artistic rendering on the right-hand side there of what a bacteriophage might look like infecting a bacteria. But really what it looks like in the lab is something more like what's here on the left. What I'm showing you here is there's a lawn of bacteria that's growing on the agar surface. And then the researcher has spotted virus on top of that. And you can see those those darker zones are zones of killing, where the bacteria aren't growing anymore because they've been killed by the bacteriophage. And it's also really interesting here, you can see there's a couple different ways that the actual plaques can look on the bacteria. So the top number 1 there, you can see that there's a halo around the edge, where there's a darker center where all the bacteria are dead. But along the edge is probably where they've become a lysogen, which I'll talk to you guys about. And then you can see there's even cloudier ones or clear plaques, which has something to do with how the viruses are growing. So I always like to talk about this, especially when I'm in an audience of people who might not know a lot about viruses, and we're talking about these viruses that infect things other than us humans. So how many viruses are there out there in the world? They actually have an estimate for that. So the estimate for the number of tailed bacteriophage themselves is 10 to the 31st actual physical particles that are out there. That's not different species of virus, but it's different individual viruses. So down at the bottom, I show you what that number actually looks like if you write out 10 to the 31st. And there's some really good like anecdotes about how big that would be if every bacteriophage was a beetle and they were on the earth. There would be 6 miles deep of beetles. So, and actually if you put them end to end at their actual size, they go way out into space, light years into space. So it's a huge part of the actual biosphere that's all around us. When it comes to the number of different virus species there are out there, I couldn't find a good estimate for how many. I don't even think we really have a good handle on how many different bacteria there are out there, so you'd have to know that in order to estimate how many different viral species are out in the world. So, but if you're talking just about vertebrates, the latest estimates are that there are up to 3 million different species that infect vertebrates of viruses. And so right now the number known to infect humans is 219, but I'm sure we're still discovering viruses all the time. And so these 2 pictures here, the first one is a transmission electron microscope picture of a phage. So this is a phage that infects a marine bacteria. And you can actually see here the tail fibers from the little like landing pod that stick out. And those contact the bacterial surface and can help tell it which bacteria it's infecting and whether or not it can actually get in. And the other picture is just a picture of seawater that they've stained with a stain that'll show you DNA. And so these larger dots that you can see are bacteria in the seawater, and the smaller dots that are sort of in the background are all the different viruses. So in 1 milliliter of seawater, no matter where you collected it from, there's over 1 million viral particles in every mil of seawater. But most of those are going to be viruses that infect bacteria that are also living there. So bacteriophage are a little different than the way we think of viruses that infect our cells that might kill our cells automatically. So there's 2 types of bacteriophage. I'm going to tell you about temperate bacteriophage, which they have 2 different, basically like life strategies that they can undertake. So the first thing that a bacteriophage does is it has to adsorb to the surface of the bacteria. So it has to find a bacteria that has the right receptor. Forget what the steps on this animation are. And then the bacteriophage is able to inject its genome, and if it decides to undergo lytic replication, then those genomes are replicated, and progeny phage are made, and then it lyses the bacterial cell and lets all those phage out into the surrounding environment. Depending on the actual phage that you're talking about, usually they have a sort of a set number that they're releasing. So some phage might release 10 particles, but a different phage might release 100 or 1,000 different phage particles. The other strategy is called lysogeny. So this is that the bacteriophage injects its genome, and then the genome is incorporated into the host genome. And now it can replicate. Every time the bacteria replicates, it replicates this phage genome as well, and basically the bacteriophage shuts down all its gene expression and just exists there, and the only thing it's making is a single protein, which is a repressor protein. And basically the job of that protein is to prevent the infection of other phage and block that so that the cell stays alive. It's trying to stay silent and not let the host cell be lysed. But at some point, this bacteriophage that's incorporated into the genome can come back out. So we don't know exactly how or why bacteriophage decide to do this, but it probably has something to do with stress on the cells. If the bacteriophage decides basically that I need to get out, this is a bad environment, maybe it thinks the bacterial cell is going to die, then it would go back into the lytic cycle and the genomes would come out and it would replicate and make progeny phage and lyse the bacterial cell. So this is for temperate bacteriophage. Basically, the other type of phage are virulent phage, and all they do is the lytic cycle. They don't do this incorporation into the host genome. So this is a slide that's based on a database that the HAFL lab that I worked in was creating, and basically their goal was to use a single host, and they've now expanded to a few other hosts as well, but to find as many bacteriophage for that one host as they could. And this is a bacteria that lives in the soil, so basically high school students, college students in some, there's a program through HHMI, Howard Hughes Medical Institute, that's put this program into different undergrad universities, and they do this as a first-year biology project. You go out and collect soil samples, and then you can filter those samples and just get whatever viruses might be in there, and do this plaquing on the bacterial lawn and see if you can get phage. And if there are phage present, then you can purify them, and then we go all the way through to sequencing them. And at this point, for mycobacteria, when I pulled this off the internet a couple days ago, they're up to 1,300 different phage that infect mycobacteria, which was the host that we chose. And they've now expanded to all of these other closely related bacterial species. And you can see the map in the middle there is just the locations in the US where they found some of these phage. And the colors tell you basically what genus group they fall into. So if they're related to each other, they're all the same color. And so the bacteriophage that I actually studied when I was at the University of Pittsburgh doing my work for my PhD was called BPS. One of the nice things about this program, and one of the ways that they get students to be involved in these projects, is that they let you name the phage when you find it. So you'll see if you ever go onto the website and look that they all have really sort of weird names. And it's because the high school and the college students that are finding these phage get to name them basically whatever they want. There are some restrictions. So this is an electron microscope picture of the phage that I was working with, that I didn't take. And the boxes here represent the genes in the genome. So you can see the ruler along the bottom sort of each of these panels basically shows you the position in the genome that we're talking about. And each of the colored boxes is a different gene that we've annotated. And we haven't necessarily been able to say for sure that all of these things are made. So a lot of basically the way a bacteriophage genome is organized is what we call the left arm is structural genes, at least for these mycobacteriophages. And the right arm are these genes that we don't really know what their function is in a lot of cases. So you can see the head and tail genes, so that those are the structural genes that make up the physical particle that the bacteriophage makes. The lysine genes are right in the middle, and those are the genes that are involved in actually lysing the host cell. And the immunity genes that you see there are the genes that are involved in whether or not it's able to incorporate into the host chromosome. And then you can see all of these really small genes, especially down here. We don't know actually if these genes are functional, if they're made as proteins, and then if they're actually made as proteins, what they do. There's only a few that we can predict functions for, which is mostly for like DNA replication of the bacteriophage. But that's actually a really interesting thing about finding all these phages, and a reason we want to go out and find out what the diversity is that's out there is because there's a lot of genes here that might be useful for making tools to do genetics in these organisms, or that the bacteria might be using to control its host. And if we knew what those genes were, we could use them maybe as antibiotic targets or something like that. And the reason we decided to use this particular phage, so basically out of those 1,300 phage that I was talking about, I spent my entire time studying like 3 different aspects of this one bacteriophage. And part of the reason we decided to use this one is that it's actually very small in terms of the genome size compared to a lot of the other phage that we found. So this cluster, it's actually not the actual smallest, but it's only by a few base pairs different. So this cluster, which would be the related phages, are all about 40,000 base pairs with about 60 or so genes. And you can see the average is a lot larger than that, up to like 75 or so. And we have some that are well over that, even up into the 100, 130 range, something like that. So we decided to study this one because it had this small genome. And if you're going to put in all this work to study one thing, you want to make sure that you study the sort of the easiest member to start with, as opposed to studying something that's the largest one that we have already. And so I'm just going to tell you about one project that I was working on, which was to look for promoter regions in this in the genome. So all of these genes have to be transcribed somehow if they're being used. So we know that the head genes and the tail genes, things like that, have to be made because those are what actually the proteins of those genes is what's actually making up the physical particles. And we don't know actually if the rest of these are expressed or not. But you could imagine that this phage is actually, when it's in the bacteria, it's circularized, would actually only need 2 promoters. It would need a promoter that drives the 2 immunity genes on the one strand, and then it needs at least one other promoter that could drive all of the gene expression of all of the other genes. We didn't know if that was true or if there were other promoters there. So one of my tasks as a PhD student was to go and find out. And one of the ways that I did this was actually just to take the genome, cut it up into as small pieces as I could, sort of was manageable, a couple hundred base pairs long, and see if those gave us any promoter activity. And so to do that, we were using a plasmid that had expressed an M-cherry gene. So M-cherry is a fluorescent protein that basically glows red in a fluorescent microscope. And anything that was a potential promoter, we could either clone in itself, or we could just put in these random fragments and see what gave us red under the microscope. And we tested them in both the bacteria itself and then also in the lysogen, so it's expressing the repressor protein. So if the repressor protein is acting on these promoters, we would know. And one thing that actually made this project a lot easier for me is we didn't actually have to use the microscope because our M-cherry gene expressed so well from some of these promoters. You can actually see it turns the bacteria pink to your naked eye. So you can see HSP60 is a positive control promoter that's from the bacteria itself. It's not a viral promoter, it's the bacterial promoter that's always on and we know expresses at a high level. And then you can see a plasmid that's in the bacteria that has no promoter. So we either get pink or we can get like this sort of yellow color that the bacteria naturally is. And sometimes we got something, usually we got something that was in between that wasn't quite this bright, but maybe you could actually see with your naked eye. And so, long story short and kind of cut to the chase, after all of that work of screening probably thousands of different colonies of mycobacteria with different pieces of the genome in front of that M-cherry, I found that there were 6 promoters in this phage. And so it's more than the 2 than we expected, but there are the 2 that we expected to be there. So the genes that the promoters that express the genes that are on the other strand, and the promoter that expresses the genes in the right arm. So you can see this is overlaid on a graph that's showing gene expression. So this is an actual sequencing, RNA sequencing that we do, where you can see what RNAs are actually made. And you can see it makes sense where the location of some of these promoters are because it corresponds with where there's high levels of gene expression in the green. So we have the 2 promoters that we expected, and there's also a promoter that's right in front of the structural genes, and those are the ones that are getting made at this super high level. So the the head, the capsid genes, and the tail genes are being made at a much higher level than the rest of the genes in the genome. And we could go through and quantitate how much expression there was, because we could look at how much fluorescence we were getting from these plasmid constructs that I was looking at. So you can see the HSP60, and we had one other promoter that was just as strong as HSP60, which is in front of gene 55. But there are also a couple of these that are regulated by the lysogen. So you can see PR, P6, and P55 all have no expression or very low expression in the lysogen, which means that when the repressor protein is present, it's able to shut down gene expression from those promoters, which makes sense in a lot of cases because you don't want, as the phage, it doesn't want its genes to be expressed. It wants to be able to replicate in the host chromosome without the host knowing it's there. And you can do things like look at the sequence and decide what a consensus promoter looks like in these organisms, in the BPS phage, and do a comparison and see what bases are required, what bases are most important. And why is this a big deal? Why, other than just to study the virus itself, would you want to know about these phage? So I was talking to you guys about mycobacteriophage. These are all viruses that infect Mycobacterium. And the most famous member of the genus Mycobacterium is Mycobacterium tuberculosis. So that's the bacteria that a lot of these will infect. We actually didn't isolate them on tuberculosis, so only some of them actually infect tuberculosis bacteria, because we didn't want to use a pathogen to do all of this work. But we've developed tools using these bacteriophage for TB research and for potentially diagnostics for TB. And you can see this is the latest report from the World Health Organization, just to give you an idea. Even though we don't think of TB as being a major problem in this country, in a lot of developing nations, it's very important, especially in relation to countries that have high rates of HIV have particularly bad tuberculosis problems, and part of that is because when your immune system is compromised, you're not able to fend off the bacteria as well. And another thing that's not necessarily related to HIV is the fact that multi-drug resistant TB has been growing in the world. So the incidence, you can see that's the graph there on the right. There's a high incidence of multi-drug resistant TB, and we need new drugs to fight this, and we're working on that obviously, or other people are. But that's also a growing concern, and as that starts to spread around the world, that could become a major problem here in the United States as well. And so I put the numbers down here at the bottom. There were 10 million new cases of tuberculosis in 2015. So even though we don't hear a lot about it here, it is a very important global disease. And so using these phage, we were able to develop tools. So the HAPFA lab has been a major contributor to this field of using phage to develop tools to use for genetics in the bacteria, including transducing phage and using these, finding different plasmid components so that you could actually make plasmids that replicate in the bacteria, as well as making diagnostics for tuberculosis. So one of the things that the HAPFA lab and some of its collaborators have been working on is making fluorescent bacteriophage, so that they would basically infect the bacteria and fluoresce, and that would tell you that the bacteria is present. One of the problems with working with tuberculosis itself is that it's really slow growing. It's part of what makes it a good pathogen too. E. coli doubles in 20 minutes. So you get an E. coli colony on a plate in one day. Tuberculosis doubles every 24 hours. And so you get a colony on a plate in a month. So if you had TB and you had drug-resistant TB, you would want to know that before a month later, if possible. And then the other thing, so this is one of the major accomplishments recently, was the HAPFA lab and Julia Van Kessel, who was a grad student, made a recombinering system in mycobacteria, which basically allows you to make genetic changes as you desire. You can make them at the spot you want, and you can make unmarked changes, which means you could replicate exactly what's happening in other strains and test to see if it's more virulent now or something like that. And then just to throw in, I'm not sure that we're ever necessarily going to use this for tuberculosis, but phage therapy is sort of growing interest in this country as well as around the world, partially because of drug-resistant bacteria. So phage were initially discovered in the 1910 decade, and they were investigated for therapy at the time, but when antibiotics came out in the 1940s and got marketed by the pharmaceutical companies, it just died down in the West. And part of that was because antibiotics work so well. But these phage were still used for therapy reasons, so to kill bacteria in Russia, Georgia, as in the country Georgia, and Poland. And so we're also using some bacteriophage preparations in food safety to be able to kill bacteria that are on equipment. And like I said, with the rise in drug-resistant bacteria, we're definitely considering this as a potential use for phage in the future, especially as it becomes harder and harder to develop new antibiotics. And like I was talking to you guys about the fluorescent bacteriophage, we're also thinking of using them for diagnostics for a lot of different bacteria that are hard to culture for one reason or another in the lab. So I just put a couple of graphics up here. The most famous phage institute that uses phage therapy is in Tbilisi, Georgia. And they have a couple of medical groups that are outcroppings of the original institute. And one of the things they do is treat drug-resistant infections. And so that's definitely something that scientists are thinking about and want to use. However, it's very difficult. There's no sort of regulation structure in place to use phage therapy. And from what we can tell, it's going to be much more effective on things that are topical. So if you could just put a phage solution on a wound, maybe that had a staph infection, that might be the sort of thing that we may eventually use this for. And so even here in the United States, people are starting to think more about this and starting to go back to this research that's been going on in Eastern Europe for basically the last 100 years. So now I'm going to move on to the project that I've actually been working on here at JCVI. Just to give you an idea, this is some animation pictures of different viruses that infect humans. And the virus that I've been working with mostly is herpes virus. And here's an actual electron micrograph picture and another animation of what a herpes virus looks like. They're large double-stranded DNA viruses. They're enveloped, so they have a outer shell that's was derived from the host cell that they came from. And everybody thinks of herpes simplex 1 and 2 when I say herpes viruses. However, there's a lot of other diseases that you've heard of that are caused by herpes viruses, including chickenpox, mononucleosis. Epstein-Barr virus is known as a known cofactor in some cancers, and so is Kaposi sarcoma-associated herpes virus, which was discovered in HIV patients. Can also cause some forms of cancer. So, and you can see in the far right-hand column of this table that the prevalence of a lot of these viruses is very high. So they've done some recent studies on HSV1 and 2 that say that over 2/3 of the globe is infected with one of these two viruses at least. Before the chickenpox vaccine was available, over 99% of the US population was infected with chickenpox at some point in their life. And as I'll tell you about in a second, one of the interesting things about all of these herpes viruses is that they can replicate in your cells latently after you've had an initial acute infection. So like I said, they're large double-stranded DNA viruses. So when I was talking before, the phage was 40,000 base pairs. These are 120 to 230,000 base pairs, so they're several times larger than that. They can be difficult to work with, which part of the reason is because they have this high GC content, which just means there's a lot of G's and C's as opposed to A's and T's in their genome, which can make it really hard for them, for us to work with them in the lab in terms of making changes, making mutations. And they're important global pathogens, not just the ones that you guys have heard, the things that you know about, obviously, but HSV2 can predispose you to HIV. I think the statistic is something like you're 4 times more likely to contract HIV. EBV and KSHV, as I said, can cause cancer, and HCMV is also associated with cancers. And they persist in the host after the initial infection. So depending on which herpes virus we're talking about, they can live in different cell types. Most of them infect through epithelial cells, but then, for example, HSV1 lives latently in your neurons. And that's actually part of the reason that there's a characteristic patterns of reactivation. For example, when you get shingles, usually it reactivates and you get a band of that's a rash. And that's because it's all of the cells along the pathway of the single nerve that was infected. And then down at the bottom here is just a schematic of what the overall sort of architecture of the genome looks like. So it's one long linear piece of DNA when it's actually in the viral capsid. And it has all of these blocks here that I'm showing you are repeat regions, which is also part of the reason that it makes it really hard to work with in the lab, because those things can recombine and switch their orientation, and they can recombine and fall out. And so basically you end up with something that's not what the original virus was at all. And part of the reason we decided to work with this at JCVI is because we have a lot of experience working with synthetic genomics. And so since 2003, you can see all the papers that I've highlighted here in red are things that were actually done at JCVI. The first thing they did was to chemically synthesize a phage genome. This is a rather small phage genome. I think it's under 10,000 base pairs. But we've gone from these small, just single genes, making up a synthetic version of the single gene, to making these small viruses. And then JCVI was involved in the synthetic cell. If you guys have heard of the synthetic cell, that's when we synthesized a bacterial genome from chemical synthesis the whole way through. These bacteria are very small. They're the smallest free living organisms on the planet. I think they're about, the smallest is 500,000 base pairs. So still very big compared to what I'm talking about. They've also done the mouse mitochondrial genome from synthesis. And we wanted to do this then with these large double-stranded viruses like herpes virus. And here on the right-hand side, you guys can see, basically this is just showing sort of the different size ranges that we're talking about working with. So we're talking about single genes that are between 100 and 1,000 nucleotides long, up to bacterial genomes that are 1 million base pairs long. So this is just a figure from the paper where they published the first synthetic genome. And it just basically is showing you how the strategy that they employed, because we sort of co-opted this to use with the herpes viruses. So what they did was they started from chemical synthesis. So you can basically buy oligos from a company where they've chemically synthesized small, I think they started at maybe 100 or 1,000 base pairs long. And then you can put those together. They developed an in vitro method at JCVI to put those together called Gibson assembly. And eventually those pieces get big enough, you start putting a bunch of pieces together, you get to like 10,000 base pairs long, and you have to move into yeast. And basically then you let yeast do the hard work of putting the things together, just because it's so hard to use and manipulate those DNAs in the lab, like in a test tube, because the second you put a pipette in and pipette up, you've sheared a bunch of the DNA, and it's now broken and it doesn't work anymore. So you see they started basically from the inner ring here where they had these tiny pieces that they synthesized chemically, and then you put them together into larger and larger fragments. They went up to, I think, thirds of the genome and then assembled the whole bacterial genome. And most of that end work then from 10,000 base pairs was done in yeast. Okay, so the herpes virus project that I'm going to tell you about is a collaboration between my PI, Sanjay Vashi. So he's the one who thought of this project and got money for it. And so we're at J. Craig Venter Institute. And then we have a collaborator at Johns Hopkins who's a HSV1 expert. His name is Prashant Desai. And then he had a lab technician who helped me a lot with all of this, whose name is Peter. And so just wanted to give credit to everybody who was involved in the project. So basically, you can see that this is a lot like that picture I was showing you before of the chemical synthesis that we did for the synthetic cell, only this is for HSV1. And instead of using a linear genome, we used one that's already a circle. It's much easier to work with circular DNA in the lab. Anything that's a plasmid we have is circularized DNA. And so we just took a strain that already, they have this these strains that are already circles that you can replicate in E. coli. They're called bacterial artificial chromosomes, so that's what the BAC at the end there stands for, the BAC. And we basically just then added a sequence so that you can use this now in yeast as well. So basically now this is one really large plasmid that you can put into yeast and E. coli. And then what we did was designed regions where we could look for where we made overlaps between these pieces. So we divided the genome up into 11 fragments. And then each of these J's is the junction between those fragments. And they have very short overlaps of 80 base pairs, sometimes a little more, between the two fragments. And that's where we're going to basically stitch these pieces back together. Is there anything else I needed to tell you before we move on, so I make sure that you can see one of the things we did was bury the repeat regions. So where I was showing you before the linear strand and the blocks were the repeat regions, here they're these green and purple boxes. And we put those inside of the middle of a fragment so we didn't have to worry about them or manipulate them just yet. And like I said, we used yeast for basically to be our workhorse, to do most of this work really for us. And we use this process called transformation-associated recombination, which basically just means we take a natural yeast strain. This is a strain that you can get DNA into really easily, and we let it do the recombination between all of these pieces for us. So this technology was originally developed at the NIH in the 1990s, and it was to clone parts of the human genome that were hard to sequence, including full-size genes with regulatory regions, things like that. So they were actually originally using it to clone pieces of the human genome. And basically all you need is to have these hooks that have, they're basically just sequence that is similar to the piece that you're looking to assemble or clone and that you're looking for. And so we use this to both do the cloning and the assembly that I'm going to tell you about. So instead of chemically synthesizing these genomes, we decided to clone them from the existing yeast strain because that would be a lot cheaper. And we figured a lot easier to parts that we needed to get all these HSV1 fragments. And there's no real reason that we had to go back to synthesizing them from oligos. We knew that that should work. So we decided to clone out the pieces using this tar cloning method. It's really easy. Basically, you just take the vector that has homology that we've added by PCR. So we just added that sequence on, and then co-transform that with the viral genome that you're interested in into yeast. And then you get this plasmid, and then you can transfer it to E. coli, and you can check to see that the plasmid is actually what you expected it to be. And then once you have the plasmid, we've also put these PME1 sites, which are just restriction sites, so that you can go in and cut out the fragment later when you want to. So you can digest with PME1. You can get rid of all that vector backbone that you didn't really want. You only were using it to replicate it in yeast and E. coli. And then we've got all of these overlapping fragments. You can pool them all together and just retransform them into yeast. And so all of those fragments have small overlaps to the neighboring fragment. And in yeast, it'll put all 11 fragments together at once back into the genome of the virus that we started with originally. And then you can check by PCR, basically. So you see the little arrows there just indicating where you might put two primers. You can check by PCR to make sure that the two junctions have actually become one, fused together, mended together. And you can see the PCR down here at the bottom has every one of the junctions present. So we figured we had a full genome here. And then it's a little bit complicated because you still have to go back into E. coli. We go back into E. coli just so we can make lots of DNA. E. coli is our DNA factory in some ways. And then you can transfect that DNA directly into mammalian cells. So you just put the DNA into mammalian cells, and the DNA itself is infectious. So just by putting the DNA in, the cells will replicate the DNA, will transcribe the genes, and make you virus. So we called this COS, which is the strain that I was using, YA for yeast assembled. So you'll see that on a couple of my slides. That just means that's the one that we actually stitched back together. And then something else we wanted to show. So over here on the right, you can see this is actually what it looks like in a plate of cells where you've transfected the genome. You can see, so the purple basically it's a crystal violet stain, and it's basically just showing you that there are cells present there. Anywhere that there's not cells, you get you can see through to the bottom of the plate where you get sort of these white spots, and that's because those are viral plaques where the virus is replicating and killing the cells. And then we did a growth curve just to show that this behaves the way we expected it to. And if you leave that sequence in, the back YCP sequence that I was telling you about, basically the sequence that allows it to be a plasmid, you actually, for some reason, the virus doesn't grow as well. So that's that sort of bottom line here, which is the regular COS-YA. But we thought about this ahead of time and made it so that there are sequences there that we could clip that out. So we removed that, and it grows just like the wild type strain of the virus does. So basically, we went from DNA in a test tube, we cloned these parts, we put them back into yeast and reassembled them into a genome, and then we're able to reconstitute a live virus from that behaves exactly like the wild type virus that we expected. And since then, we've now sequenced this, and we've showed that basically it's 99% identical to the template DNA that we were using to build this from. And so that's great, but basically we've just remade herpes virus. So we wanted to show then, the whole point of this was to develop better tools to do genetics in herpes viruses. So one of the things then we wanted to do was take these fragments of HSV1 and edit them in some way, make either gene deletions, make insertions. One of the things we did that I'll show you guys is we put fluorescent tags on some of the proteins. So now that you have these smaller pieces of herpes virus genome and fragments in yeast and E. coli and in the lab in a test tube, you can use a number of different methods to make whatever changes you want on those plasmids. And so one thing that's really in the news right now that everybody's talking about is CRISPR. Maybe you guys have heard of. And they're talking about using it to do edits in mammalian cells and maybe eventually in like human cells. I think they've actually done it in human cells, just in cell culture in the lab. But we used that system as well as like PCR and restriction cloning, sort of all these things, these tools that we already have in the lab to make changes. And so one of the things we did was we made a series of fluorescent protein fusions on VP16. So VP16 is a protein that's really important for herpes viruses, and it is part of the actual viral particle. It's in the tegument, so it's the space that's between the capsid and the envelope, and it's an important transcription factor for herpes virus. And one of the things we just wanted to show is that it was easy to make changes now, where before it would have taken you months to make a bunch of changes. Now we can do this in a few weeks. So we made different fluorescent protein fusions. So we basically made just different colors of this protein fusion. So here I'm showing you the sort of genetics that in the gel here that shows that how efficient this was, that we're getting changes 75% of the time. We're getting a positive change. And then you can just see that by PCR over the junction of where the gene and the fluorescent protein that I've inserted are. And the proteins get made, which is important. So the left-hand panel here is a Western blot that shows that the proteins are being made. You can see that the untagged version is much smaller than the tagged version, which is exactly what you would expect because we've, I think, more than doubled the size or about doubled the size of the protein. And the viruses grow fine, which is the panel here on the right, just showing you that now we haven't like compromised the ability of the virus to grow. And then under the fluorescent microscope, you can look at these and you can see we've made a bunch of different colors. So basically now you can use this as a tool for herpes viruses to tag different proteins throughout the genome, or to just use to show viral replication if you needed to in a particular mutant cell line that you were, or strain, herpes virus strain that you were making. So we made different colors of blue. Venus is actually, it looks green here, but it's actually a yellow fluorescent protein, and two different red fluorescent protein fusions. And these are all on this one protein. But we're just trying to show that you can make changes more quickly. So since then, what we've also done is made deletions throughout the genome in other genes, particularly in some of the tegument genes, which is what our collaborator at Johns Hopkins is most interested in. He spent his whole life basically studying these genes that are in the tegument of HSV1. And so we're going through and making a bunch of deletions in those genes, and they're in different places throughout the genome. But I won't show you guys all of that. So we've got a paper coming out, hopefully soon. It's out, it's submitted and under review right now. So if you guys are interested, you could always look that up and see sort of all of the things that we've done and what the sort of future plans are, things that we think we can do with this system. So I want to tell you guys a little bit about some of the other viruses that I've been working with. So this was about 2 years of work for me. I started at JCVI in 2014 in like September, and that's really, some of the work had already been started, but that was really when we got started on this project to do synthetic herpes viruses. And so that's what I did basically for my postdoc, which I'll tell you guys about postdocs in a little bit if you're not familiar. So that's what I started doing. But since then, I've moved on to some projects working with other viruses. So one of the ones that's very in the news right now is Zika virus. So at JCVI, we are a Genomic Center for Infectious Diseases. So the program is called GCID, which is through the National Institute for Allergy and Infectious Diseases at the NIH. They basically set up a couple of sequencing. We're basically like a sequencing core that's doing sequencing for different viruses. So originally the program was set up to sequence things like influenza and rotavirus, respiratory syncytial virus, West Nile, chikungunya. So some of the existing viruses that are out there right now that we didn't know a lot about how they were changing in the environment. But after Zika, the Zika outbreak that happened, we also started studying and sequencing Zika virus isolates. And so just to give you an idea, so Aedes aegypti, I'm showing you the predicted range here, is one of the 2 mosquito hosts that can actually transmit Zika. So you can't get Zika from another person, you have to get Zika from a mosquito. So these are the places where you could potentially see Zika virus spread. You can see that obviously there are parts of the United States that are that could be involved, and they have had some cases in Florida. And then they also have cases in the United States sometimes from people who've traveled to other places and then come back. And there's a picture, just sort of an artist rendering of what the Zika virus looks like. So basically, this project is all still ongoing, so I don't really have very much I can tell you about it. And I actually just joined the project about 2 months ago. But they have published one paper, and basically we've set up about 10 different collaborations with people from all over the globe, especially in South and Central America. So we've got collaborators in Colombia, Honduras, Nicaragua, Barbados, Mexico. A lot of those researchers are based here in the United States, but have labs or have collaborators themselves in these countries. And they're sending us samples, and we set up different projects for each one of our collaborators where we're addressing a specific scientific question. Maybe we're looking at transmission of the virus in that country, or we're looking at different phenotypes. So we're also trying to, correlate disease severity with the strain of Zika virus that you have and see if that's actually a major component, or is it mostly your own genetics that plays most of the role. So that's kind of what's going on in that project right now. I don't have a lot of data to show you, not that you guys need any more, because I showed you a lot of other stuff already. And then the other virus that I've been working with recently is enterovirus D68, which has also had a recent outbreak in 2014. Before 2014, it was pretty rare to see this particular enterovirus species. But in 2014, they had a major outbreak that was, I want to say like 1,500 cases in the United States, which was, I think they had seen like 500 cases, maybe not even that many, maybe like 150 cases in the past like 30 or 40 years before this particular outbreak. So there was a major outbreak, and so it coincided, and we can't say for sure that it is the cause, but it coincided with an outbreak of paralysis in children. So the actual clinical term is acute flaccid myelitis, but it's basically, this virus is related to polio. So it's the same thing you would think of with polio paralysis in children. In 2016, there was another outbreak. We're not sure if it was as serious as as large as the 2014 outbreak. We don't have the actual numbers yet. And there were also increased acute flaccid myelitis cases that year as well. So we're not sure, we can't say for sure that that is the cause of this paralysis as well. But normally, if you get enterovirus D68, minor symptoms would be, you would just have basically a cold. Most severe symptoms are more things like wheezing or difficulty breathing, which can still be fatal depending on the person and the child and how young they are, things like that. But we think that also it might be causing these polio-like paralysis cases. But the CDC is not confident. If you go look on their website, I was looking at it today actually, they don't talk about these paralysis cases at all. So they're not confident that this is real. But we have, one of the PIs at JCVI that I've been working with, Richard Sherman, he actually works on the West Coast at our La Jolla campus, has done some sequencing and phylogenetics basically to see how related these strains are to each other of the virus, and has seen that he thinks that there's one particular subclade that's actually involved in these paralysis cases. And just 2 weeks ago, a group from the University of California, San Francisco, and the University of Colorado published a paper that showed paralysis in mice. So I'm going to show you guys a picture of a mouse. Don't freak out. So you can see this particular mouse has hind limb paralysis. I'm not sure how often this is fatal or if it's just a complication. And even with polio, sometimes people that had paralysis would recover, but usually only partially, and that you wouldn't get maybe full development of the limb back. So it's interesting. We think that it might be involved in this respiratory virus might be involved in this paralysis cases, but we're not entirely sure. And basically we need to do more surveillance out in the community and find out if people that have paralysis have this virus. And I think part of the problem, the reason they're really not sure yet, is usually by the time you're presenting really severe symptoms, the virus might actually be basically gone from your system. So that's something that definitely the field in general is still working on. But at JCVI, we've been basically trying to do this to develop a cell culture model where you could see this difference between these different strains of the enterovirus D6D8, basically lineage. So in HeLa cells, you can see we can grow. So we've got the first picture here is a mock, which basically is no virus is added. And you can see the cells in the bright field, and DAPI stains for the nucleus, so you can see the nucleus in the first set of fluorescent images there. And then the last is staining for one of the viral proteins, VP1. So you don't see anything in the mock. There's no virus there, so we don't expect to see any viral protein. And then this historical strain that's from, like, I think the 1960s grows well. This B2 strain grows well, and this B1 strain grows well. And then we've got a neuronal cell line that basically shows that you don't get growth of any of these these other strains except for the one that is the B1. So we think basically that maybe we have a cell culture model that might work for this. And that would be very preferable to having to do a lot of this work in mice because it's expensive. And if you don't have to kill mice, you don't have to kill mice. So we're working on that. We're developing this cell culture model. And then we hope to be able to basically go in and do program in some of these genetic changes between the different groups and see if we can pinpoint what region might be responsible for its ability to grow in neurons, which would then also probably give us at least a hint of what might be responsible for the paralysis. So it's nice now to also have a mouse model because if we see changes, if we see differences in the cell culture line, we could test it then in the mouse model. So I just wanted to put this up because I'm done with the science part. And just say thank you to everybody involved in the work. This is a short list, but I tried to talk to acknowledge all the people at the University of Pittsburgh that were directly involved in helping me, and at J. Craig Venter Institute, and then our collaborators at Hopkins, and Claudia, who's in our audience, has been my intern at JCVI, and she's been a big help she's been helping me with the HSV1 project. So you guys too could eventually, maybe someday, intern at JCVI if you'd like. Okay, so now you guys feel free to ask me questions as I go through this. I just kind of wanted to tell you how I became a scientist, and how I made that decision, what I decided to do. So the University of Akron is where I got my bachelor's degree. I'm actually from Akron originally, and so I decided not to go very far away for college. And I did a biology degree. I don't know if now they do, but at the time they didn't have actual specializations. You could get an area of specialization. So I got an area of specialization in micro, which was basically like 3 or 4 upper level classes, but we didn't have an actual like microbiology degree or biochemistry degree that you could get. So that was, in some ways it was nice because it allowed me to have kind of to study a broad range of things in my bachelor's, because there was no one particular program that I joined other than just biology. And then I was a grad student at the University of Pittsburgh. I went straight from my bachelor's to my PhD program, which is really the way that it's done mostly now. Most people don't go on to get master's degrees and then get bachelor's degrees, although there's still a fair amount of that happens as well. If you decide to go get a master's, what a lot of people do is they end up working in the lab as like a research associate, and then don't go on to get their PhD. And you can work in like an industry sort of, instead of an academic role doing that. Oops, wrong way. And then I know you guys are probably interested in this. So grad school obviously is really long. And then after you're a grad student, my PhD was 5.5 years. So I did 4 years of undergrad, 5.5 years of PhD. You're still really not a scientist yet. You are. But you still have additional training that you need to do at that point. So you go on usually to a postdoc, depending on what exactly it is you want to do. If you want to become a professor, it's basically required now that you have a postdoc, which is additional research under another scientist. So I did my postdoc for about 2.5 years, which is actually really very short. It was about 2 years, because it was about a half a year ago I got promoted to being a staff scientist, where I'm definitely more independent. I'm able to move on to more projects and do more things now. But generally speaking, you could be a postdoctoral researcher for, usually it doesn't go for longer than 5 years or so. And then you would, at that point, maybe apply for academic jobs to be a professor, or go to a place like JCVI, where we do have like a professor structure, but you're not necessarily teaching classes. You can also work in industry after doing a postdoc. Maybe like work at a pharmaceutical company, something like that. So you're still, the nice thing about grad school, one of the nice things about grad school is, as opposed to med school, you get paid to be a scientist in grad school. So depending on where you go, they might pay you a different amount. But you do get some sort of stipend in the sciences usually. And they cover your tuition and things like that too. So it was nice to do that at Pittsburgh, at the University of Pittsburgh, because Pittsburgh is a nice, easy, cheap place to live. Because you don't get paid a whole lot more just because you are a postdoc in Boston compared to Pittsburgh, but your cost of living is a lot different. So. And then one of the things I love about science is you really do develop close relationships with people, especially in your lab or in your class at school. In my PhD, our whole class was 13 students. So you get to know each other very well. You end up joining different labs and then making friends outside of just your class after that. These are just some pictures of my lab mates at Pittsburgh up in the right. And this is me at my defense on the top. And one of the nice things too about being even a PhD student is that I did get to travel a fair amount for work and go to conferences and meet people and go listen to interesting talks. So the picture down there in the right is me and my friend Val, who is another PhD student in my lab. We got to go to a conference in Belgium, which this was pretty fancy because my boss did have a good amount of money to send us to conferences, so not everybody gets to go overseas, but we got to go to a conference in Belgium on the topic of the conference was viruses of microbes. So it was bacteriophage, but it was also viruses that infect things like archaea or yeast, single-celled yeast, things like that. So you get to do lots of that stuff. So, okay, I've talked for too long. So, anybody have questions? You want to just kind of like? Mm-hmm. Yeah. Yeah, so the actual project itself that we've set up through NIH is part of why they wanted people to, us to be the sequencing core is they'll pay for the sequencing. Basically, NIH is funding the sequencing, so they pay us to do the sequencing and the analysis. And then we send those sequences back to our collaborators, and they can write a paper or decide how they want to use that information. But then also we, within I think 45 days, publicly release the sequences on GenBank, which is like the biggest sequence repository on the internet, basically. And they are the ones that have all the sequences of microbes and things like that. So that's part of why they set that up too, is for us to be able to get that information and then also release it publicly so the rest of the scientific community has access to it. But then when we establish these collaborations, a lot of times we don't just do the sequencing, we actually are involved in some science questions they're trying to answer and are part of that process too. So we're not just doing the sequencing. But we don't do a lot of growing the actual virus in the lab ourselves. We do some sometimes, but usually they're doing sort of more of the biology and figuring out exactly what question they want to ask. But just knowing the sequence can tell you a lot. Other people, I have discussion notes for you guys. If you so just some of the things I was thinking of when I was thinking about like my career and what brought me to where I am is just that like so much of, I think life in general, in your career and in other things is luck. I applied for one postdoc and it was this one, which is not very common. But usually people are sending lots of emails and cold calling people and go on a lot of different interviews, and this was actually the only place I applied. So it's one of those things like it's good to be lucky in science in general, like getting good results, and also in your career, but it's also good to like take the right opportunity when it comes along. So when I actually applied for this job, I didn't hear anything for about 4 months after I applied, and I thought, oh, they filled the position, they didn't even consider me, and I got a call after 3 or 4 months, and they were like, can you give a talk in 2 weeks? You want to come down and interview? Like, okay. And then they were like, you want to start in 2 weeks? So I moved from Pittsburgh with about 2 weeks notice, not knowing what I was going to do. I finished my PhD. I was working in Pittsburgh in a restaurant as a hostess for about 2 months over the summer, not sure what I was going to do. Not sure if I wanted to stay in science. I was thinking about doing science policy or something like that. I was thinking about sort of like changing out of the research career, and got lucky and got this call, and 2 weeks later I was working here at JCVI. So, and just like, you don't necessarily have to know exactly what you want to do, but if you're doing something right now that you love, just keep doing that and look for the opportunities as they present themselves and be ready to take it. If you get a call on 2 weeks notice to come give a talk, or and then 2 weeks later to start a new job. Like, that's sort of how I ended up where I am, is a lot of it's luck and a lot of it's, having the right skill set, but yeah.
General question.
Yeah.
Nothing to do with science, but what made you choose biology or computer science or
Mm-hmm.
maybe my kids to understand,
Yeah.
How to choose the field when they're in high school?
I got really interested in high school. I had good science teachers, which helps a lot, obviously, that were very interested. And I had a PhD teacher in high school for my chemistry. That's actually what I loved in high school, was chemistry, and thought that that's what I was going to do. And then I walked in to my first year at Akron, and I started taking OChem because I had taken AP chemistry. I was like ready to go, and I hated OChem. And I just knew that that wasn't the track probably that I wanted to stay on. So I got a chemistry minor and switched to biology and was much happier. But what actually originally, one of the reasons that I decided I wanted to do infectious disease research is like, it's always been kind of in the back of my mind, and it was because of a book I read when I was maybe 16 or 17. It was The Hot Zone, which was about an potential Ebola outbreak here, and it's very sort of fictionalized, and it's not really what science is like at all, but it's what got me interested, and so I always think about that. It was a book my grandma gave me, and I had good science teachers, and I think that's a lot of why I ended up in biology. Yeah. Yeah, they do, for sure. Good science classes helps a lot. But then I didn't necessarily go to like the most prestigious undergrad institution either. So it's one of those things that like you can still follow a track no matter where you end up at any one point. You can still keep pushing forward. So. Anybody else have questions about being a scientist or? Yeah, go ahead. Probably, but no. I mean, like I think being curious in general and like definitely being willing. I've switched a couple of these projects that I was telling you about at the end. I've really been working on for like 2 months now. There's still a lot that I don't know. They're not viruses or, even in the family of viruses that I've ever worked with before. And so I think being willing to admit when you don't know what's going on is good because that's the only way you're going to get more information. And I'm surrounded at JCVI by a lot of smart people, and so you have to be able to admit when you don't know in order to get the information that you need. So I think that's important. Caleb? Oh, go ahead. Go ahead. I think I still have a lot of like love for bacteriophages in general, just because it's not something that a lot of people are like. There's a big group of people that are working on bacteriophages, obviously, but it's not something that everyday people are thinking a lot about, and I think they're really important. And there's a lot of tools and things that we can use, like ways we can use to develop them, whether or not we ever actually really get into phage therapy, whether or not it actually ever really works as like a therapeutic that you give to people. I think just they're so prevalent out in the environment, and they're so important to like the bacterial ecosystem that's really happening all around us all the time. And I never thought I'd be working on herpes viruses. I really do like studying herpes viruses. They're really cool. So I think whatever you end up studying is the thing that you love at the time, because you start to know so much about it. And yeah.
What would you say is the downside to your career?
Oh, there's definitely pros in that like this is what I go to work in every day. I get to wear jeans and tennis shoes. I don't have to dress up. I don't have to be super formal with people. I have lots of good relationships with people, but like the hours can be really long sometimes. They're very flexible in science. It depends where you are and who you're working for. But like my employer here, my boss at Pitt, everything was very flexible. There was a student at Pitt who was Taiwanese, and he used to work on Taiwanese hours. And I don't know why he used to do it, but he used to work in the middle of the night. And he was, when you're training and learning new things, you have to do that. But you have to work on other people's schedule. But it's nice because it's flexible. But the hours are long. You just sort of like never leave work. Whenever I'm home, I'm thinking about like, oh, I should really be reading these papers that just, something that just came out, or even like when you're on a new project, you're trying to figure out what the like lay of the land is. So the hours can be long. There's not always good job security because you're on a grant. I started this postdoc on a grant that was a 2-year grant, and I didn't know if after 2 years I was going to still be here, or if they were going to be like, all right, your project's done, we're out of money for you, go find another job. So that can be a little tricky sometimes too. But it just can be, and it swings sometimes too from being really boring one week to being really high stress the next week, depending on sort of like what's happening. But now that I'm starting to do more like sort of project planning and thinking about what I want to do when I'm running my own lab and the sort of questions I want to address, I think it's just high stress all the time as opposed to being boring ever. But so writing grants and things like that, you're on a deadline. Giving a presentation in front of a bunch of people, you're on a deadline. So that's a good question though. Yeah. No, go ahead.
For high school students, they have to take a picture for some of the research work that you are doing. Is there any prerequisites, or what is the procedure that they have to?
JCVI, we don't have a lot of high school interns. We do a lot of more college internships. But for high school students, usually, as long as you can show that they're like responsible, and like that's more sort of the important thing, as opposed to them being necessarily like the smartest kid in their class. but knowing that they could come into an environment with a bunch of biology researchers and we can trust them not to like hurt themselves in the lab or to do something they're not supposed to be doing is really more important than necessarily like the prereqs, or some biology knowledge is always helpful, but the reason it's hard to do internships for high schoolers is because the biology that we're doing is sort of like on another level. And so even with college students, though, there's a lot of training and work that you have to do. So, but I think that's the only way you learn it, is you have to come in at sort of this level where you really don't know anything about what you're doing. But, so we do internships for some high school students that I'm sorry, you guys, we don't have anything open right now. So, but be thinking about it for next year. So I think we got I think like hundreds or thousands of applications for our internship openings this year, and but it's up. They posted up on the website. So if you guys are ever interested in doing something at JCBI, just look in I think they posted them in January for the summer. So we do internships, some internships for high school students. We have a specific program for community college students. So we get lots of students from Montgomery Community College, and then just some of the professors, if they have money to support an intern, will say that they want an intern for the summer, and we get college students usually for those projects. So, yeah. Yeah, go ahead. I think grades are, it's so hard because I really don't think it's what makes you a good scientist. Like when you get to being where you're actually working in lab on a specific project, like being able to learn new things is important, but not necessarily the grades that you got in high school or the grades that you got in college aren't necessarily going to indicate whether or not a person is going to be a good scientist or not. Although I feel like it's one of those things that like, you have to be getting good enough grades in order to be moving on to the next step at every point. So it's important, basically like as in like on your resume thing. But I know plenty of great scientists who are C students. Yeah. Yeah. I think it definitely helps just having, I mean, that's going to help you because you're going to get recommendation letters and things like that too, which are really important in an application. And you just need to have a sort of a little bit of everything enough that like your resume is good enough that you get the interview. That's always what I'm trying to do, is like make my resume good enough that I can get the interview, and then hopefully I can convince them at the interview that I really am smart enough to do it. So like I think that's sort of the key. But yeah, experience definitely, being able to show whether you want to go to med school or grad school, having research experience I think is really good. And then if you want to do go to med school, like these sort of like shadowing programs and things like that I think are also like really helpful to show them that that's something that you really want to do. But it's hard because a lot of times all somebody's looking at is a piece of paper, and you're just trying to make that a good enough showing that you can meet them. Yeah. Anybody else? Caleb, did you want to ask something? Mm-hmm. Yeah. Yeah, I think having doing these sorts of experiences, whether it's volunteering, whether or not that's in a science sort of field or not, but just having that sort of thing to show that you want to be well-rounded, which I know is sort of like a cliche these days, but showing that you're interested in things other than just your schoolwork is always a good thing. Having grades are important. Doing internships is great. I think that one of the things that really makes me a great scientist is that I love to learn, and I'm always looking to learn something new. I'm a reader. Like, I think that those things are actually really kind of, it's not something you can put on your resume, but I think that sort of curiosity and wanting to learn is what makes you a good scientist. And if you love that and you're good at that, I think that you can succeed. So, think that's really important. Anybody else? Did I have anything else interesting that I was thinking about talking to you guys about? I wanted to say too, just like, I think one of the things I wanted to say was, so this is sort of, in a lot of ways, I've done sort of a very traditional career path, and that I went straight from high school to college, straight from college to grad school, and then I did a postdoc, and now I'm doing basically a research job, which is not actually the way that a lot of people's careers pan out, and that there's lots of other things you can do with a science degree other than just do research in a lab. Obviously, lots of people talk about going to professional school, med school, or dental school, or something like that. But then you can also do things like become a science writer, or do consulting for science for a company, or become a patent lawyer and work in the law with and having your science knowledge really plays into that. So there's lots of other things you can do. If you love biology and you want to do that in college, I totally encourage that, and I think that that's great. But just know that part of the problem, I think, is that we don't train scientists in high school and in college. Kids that are thinking about science don't hear a lot about some of these, they call them alternate careers, which is sort of, which is not, it almost sounds bad the way they talk about alternate careers, because it's just as much of a valid career path as deciding to go into research or deciding to go into medicine. So there's lots of other things you can do, and if you're passionate about biology and that's what you love to do, I would say, I mean, you obviously want to be thinking about what your next career move is, but not to let that stop you necessarily if you're not exactly sure what your next step is going to be. So, yeah.
Also, are viruses living?
It's a good question. That I don't think, I don't have any problem with people calling viruses living things. But lots of virologists don't like that, and lots of other biologists don't like that, so I don't know. I think where they want, I was reading about this recently, and I think where they decided they want to draw the line is at ribosomes. So viruses don't make ribosomes. They don't have genes that encode ribosomes. We don't know of any viruses that has a gene for a ribosome. And instead they have capsids because they have to make this protein code. So that's sort of like where they're sort of putting the line at life, is like if you have a ribosome, you're this thing, and if you have a capsid protein, you're a virus. So, but I think it doesn't matter too much. I think, as long as you're you understand well what you're doing and why you're doing it, whether or not you call it life or not. And there's lots of things that are actually, we talk about viruses as being, among the smallest replicating organisms, but there's also like prions that are just proteins that self-replicate themselves, and viroids, which I think are just circular pieces of DNA, and plasmids themselves move between bacteria and move genes around and things like that. So there's even like sort of this like even lower class of things without ribosomes that are even smaller and less complicated than a virus. So, but it's a good question, and I don't know. I think it's more of a philosophy question than it is even a science question, whether or not you call it a living thing. Yeah.
I thought I heard from somewhere that they were talking about creating a bacterial environment, taking some sort of retaking their DNAs or something, and then start changing the colors.
On the outside of the bacteria itself? I don't know anything about that. I mean, you could do that, like that's sort of what we're trying to do with phage, with these fluorescent phage, is to be able to turn the bacteria a particular color, but that's not necessarily on the outside of the bacterial cell. That's really on the inside of the bacterial cell that's happening. But it could tell you whether or not the living bacteria is there, because only in the living bacteria can you get the phage infection and growth. But no, I'm not sure what people are trying to do with that. Yeah, Caleb. I think it depends what school you're applying to for grad school and sort of like what they're used to. I know that to some extent it's almost harder in some ways to get into some biology and medicine programs with a biology degree, just because you're one of many. lots of people decide to do that. And I remember thinking like, man, I should have done my chemistry degree after all, because at least it makes you stand out. You have sort of this different skill set, but it's related. You definitely have to have some background in biology. I don't necessarily think you have to have your degree in biology to decide to become a biologist. And obviously you can go back and take classes later if you ever wanted to change careers. But I think having some classes in biology and maybe like a minor or something like that would be a good thing. Depending on how far you want to venture from the life sciences, like engineering I think might be kind of hard to convince a biology PhD program that you were equipped to kind of hit the ground running. But that doesn't mean you couldn't go do some additional classes after you finished your engineering degree or while you were taking your engineering degree. So I think it depends how far away from the discipline you want to go. But definitely, I mean, I never thought that I would be thinking as much about engineering as I feel like I am now. Part of that's the synthetic biology and thinking about engineering new pathways and systems. And that's, I think, what a lot of the sort of, especially the people that are involved in bioenergy at JCVI are thinking a lot about how to engineer algae and things like that to grow at faster rates so that you could use them to make some sort of power to, you harness the photosynthesis, things like that. So in some ways, all of science is becoming so much more sort of cross-disciplinarian, and that like you have your training in this one thing, but being able to reach out and speak the language enough in terms of the science to talk to an engineer and get their perspective on what you're doing is good too. And that's why I think it's also really good if you guys are thinking about medicine, that you have a strong background in science and biology and maybe some research experience, because it's so important for doctors to be able to understand sort of the ongoing changing landscape of science and how it's all done. And I think that makes medical professionals better at their job too. So, yeah. I would say if it's something you're interested in doing, to keep taking biology classes while you're in high school, while you're in college, even if it's not what you think you want to do for your major, just to keep like yourself involved in it. And when it comes to actually having research experience or something like that and actually working in a lab, that's really probably college. And generally speaking, wherever you go, even if it's not a college that has a grad program in biology, there are still professors there who are doing their own research and have programs where they take undergrad students into their lab, usually as volunteers. Unfortunately, it's not very often that you get paid to do this as an undergrad, but where you could volunteer in a lab and work 3 days a week for a couple of hours and get some research experience. And actually, it's not really until you do that you really understand whether or not, you like doing science research, and where you actually can start to gain the skills that you need. Because just taking classes, and even like the labs that you'll do in your classes in college, don't really replicate what it's like to actually work in the lab at all. So it's important and it's good skills still, but it's not really the same as actually having a job working in a lab where you're working on a specific project every day, or a couple days a week or something. Yeah.
So as a young student, 10 or 15 years old, and you have this idea of what a doctor is based on what you see on TV maybe, or you have an idea of what a lawyer is. So can you give us an idea of what is it really like to be a researcher? And say you're a young student, you say, I want to be a scientist. I want to be a researcher. In your head, you think,
pouring the colored liquids from test tube to test tube, like all the pictures show, right, of like a scientist in a lab coat. So it's changing a lot for me right now. Because I've gotten this promotion, I'm a staff scientist now, I'm looking to become a professor. Basically, I'm doing a lot of writing grants, which is not anything that I have experience, and they try to train you to do that in grad school, but it's not your everyday, day-to-day life. It's more like doing research at the bench, depending on if you want to do wet lab or what we call dry lab, which is to be basically a computer scientist or a bioinformaticist. But so when I was in grad school, it was a lot of you come in every day, you have your bacterial cultures that you set up, you're doing this new experiment, you're using a new protocol. A lot of experiments that the sorts of things that I'm doing could take you like several hours. So it's not like, come in and work for an hour or two and then sit down at your desk. But there's a lot of like movement back and forth. So you spend 5 minutes working on something, set something up, go sit down and read a paper for a little while, and then go back. And it's sort of all day. And then it depends. So then sometimes you'll be there super late one night, and then the nice thing about grad school is it's really flexible too. It's like, I'm gonna read papers from home today, like take a day off. Sometimes you've got a time course where you have to take a sample every hour for 14 hours, and so you're there in the middle of the night by yourself taking samples. And so a lot of it is actually like you're actually doing things in the lab with bacterial cultures, setting up PCRs. There seems to be a lot of like using a pipette and pipetting clear liquids from tube to tube, which like doesn't always seem that interesting, but if you understand the like sort of big picture of what you're doing and why getting a good, a particular result is going to move you forward on the project, it can still be exciting, but it can be tedious in some ways to work at the bench. Some people love it, and I didn't. But it's sort of hard because that is what you have to do in order to get to the place where you're doing more writing and thinking about science like I am now. And I actually am enjoying this a lot more than working in the lab all day. But so now I'd say my time is like, some days it's like 100% meetings, where I'm having meetings about different science projects that are happening. I'm having phone calls with NIH or with a collaborator. On the Zika project, we're doing a lot of that. It's a lot of just meetings and organizing and sending emails, coordinating things. But then a grant deadline will come up where I wanted to write a grant, and I have 2 weeks to do it, and all I do for 2 weeks is write grants, write the grant that I'm looking to submit. So it's sort of like it changes every week or so. Like it seems like I'm jumping to a new thing, but now it's a lot more reading, writing papers. I have a couple of interns. Claudia was my intern for a while. I have another intern who's from Montgomery College right now. So I'm helping them set up things in lab, but basically trying to train them to do all of the lab work so that I can go and do the other things that I need to do, and they can be doing the lab work for me, which is basically when you become a professor, what you do. You write grants, you get the money to do the science, and you help a lot with the papers. And then you go to meetings and you talk to groups of people. So I've got a couple of meetings scheduled that I'm going to this year to talk about different projects that I've been working on. And that all sort of comes later in your career. So I'm just starting to do that. And that's basically once you're a professor, that's what you're doing, is doing a lot of writing. And I didn't know if I would love it, but I actually am really enjoying it. So that's good.
So on the topic of grants,
Yeah.
is there a great deal of pressure to obtain a grant before embarking on a research project?
So it depends where you're working and how sort of the money situation works generally. Like a lot of times when you become a professor, when you get your like assistant professorship job, they give you startup money. So they give you usually a fair amount of money to like enough for a couple of years worth of work, maybe to hire a grad student and get started and buy reagents and equipment that you need, things like that. But then after that, you're kind of on your own, and that like if you don't have money, depending on the academic institution you're in, they might be able to pay you to teach, but you won't have any money to actually do the research anymore. And then it gets hard once you run out of money and you're out of money for a while. It's hard to ever get money again because you don't have money to do because a lot of these grants you need preliminary data. So you need to have already generated some data that shows that like your project is a good idea that nobody else is really working on, and that like there's a good chance that it'll work. So it can get hard to get grants again. I've noticed basically that because funding has been kind of tight in recent years, it's definitely been the way that Congress has been giving us money is basically to give us about the same amount of money every year, maybe a small increase, but it's not keeping up with inflation. And there's a lot of young researchers who are having a hard time getting grants. And so it's kind of a numbers game. I just submitted my first grant where I'm like the lead PI, maybe 2 or 3 weeks ago. But I won't find out for 8 months. Yeah, fingers crossed. But I won't find out for 8 months whether or not I got it. So right now I'm like working with other people on projects, and one of the nice things about JCBI is it's so collaborative that I started working with Sanjay, and I basically was the only person working on that project directly in JCBI. And we ran out of money maybe 6 months ago or so. That grant basically ran out, and I was like, oh no, am I gonna have a job in a month or not? And there are other professors that had other money that said like, yeah, we'd love you to come work with us on this project. So that's how I got involved in the Zika project and this enterovirus project. But yeah, there's a lot of pressure. And at JCEI, it's hard because we don't teach, which could be a good thing or a bad thing, depending on whether or not that's something that you would like to do as a professor. But it also means that there's no money at the institution to support your salary based on teaching. So your salary is completely and entirely dependent on the fact that you have research grant money. So there's a lot of pressure, but the nice thing is if you're at a great institution like JCBI, like you can move around, and there's these other projects that you can work on that can help support you when you're in between grants, in between money. It's good. Go ahead.
So it's kind of a general question.
Mm-hmm.
So say like as a doctor, you know, like say things like as a lawyer, you find like justice. What is it people feel like science and society?
The really cool thing about science is that like I do a lot of learning about what other people have done, but the really exciting things that I'm doing, I'm actually figuring out things that nobody else has ever done before. And so like you're not just learning about the field necessarily, although that's a big part, but you're actually like literally doing things that nobody else has ever done. And it can be really hard and frustrating, but when you, and I think the time that I always feel like sort of the most joy is when you publish a new paper, because basically it's saying like, this is something that nobody knew about before, and I wrote this paper and somebody out there decided it was worthy of being published, and scientists reviewed it and said this was good work, and now there's this new piece of knowledge sort of out there in the world that never was there before. So that's the really cool thing. But then part of the reason I love science too is that I'm jumping around onto all of these different projects between different viruses, learning new things, getting exposed to new pieces of science. So. Anybody else? Like what? Oh. So this is the first presentation I've ever done like this. So like I give a lot of presentations actually, but mostly it's internally or for like new collaborators or things like that. So we've been trying to establish new collaborations, especially to work on some of this bacteriophage. We're starting to think about working on bacteriophage with my boss Sanjay. And so I've been doing, basically this sort of what you saw in terms of the herpes virus project and how we break it up into pieces and then reassemble it back. I've been giving a lot of those presentations, like probably 3 or 4 this year already that I've done that. But big presentations like at meetings, you might go to like a couple of meetings a year, but the more prestigious you get, the more of your life becomes that too. Like the president of our company is, I feel like she spends basically like half her time in Rockville and half her time in La Jolla and half her time traveling. So she's gone probably, I think she said this year she only wants to go to 1 meeting a month. So it can become something where if you really wanted to travel, she gets more invitations than she can possibly accept. If you wanted to travel a lot, you can go and give presentations. So I do a lot of stuff that's more local now, but sort of as you become more prestigious in these meetings and people want to invite you to come speak at their university, you might be, she's in Germany one day and she's in Barbados the next day, and then she's back in San Diego at the lab. So sort of depends how much you want to do it and how much you get invited to give presentations and things like that. Yeah. Go ahead, Caitlin.
First of all, give a quick thanks to Dr. Oldfield here.
Thank you. Thank you guys all. You guys were a great audience. That was nice.
Yes.
Have a nice discussion.
Yes. And also, for those of you who enjoyed this presentation, on March the 30th, we will have this gentleman right here, Dr. Daniel Nelson. Correct? Yes, he works in the Department of Veterinary Science with the Institute for Bioscience and Biotechnology Research. He will be discussing antibiotics, antibiotic research, resistance, and the Nelson Lab. They actually develop enzymes that act as an alternative to antibiotics, which I find rather fascinating. So if you are interested, on March 30th, it'll be on Thursday, very end of this month, in this exact same room, at the exact same time.